All right, thank you very much. I'm going to speak about metagenomics and cannabinoid response, and it's a big reach right now, but hopefully by the end of this I'll convince you that they're actually connected. Over a decade ago, we started sequencing the human genome across the river. A few people in this room were involved in building a robotic field to basically construct 2 football fields worth of robotics. It was in East Cambridge because the real estate was cheap, and that's an important point. Now, that took us several years to build the robots, but in 10 months, those robots finished sequencing 40 million E. coli clones. And we spent painstaking efforts to make sure there wasn't any microbial contamination in the human genome reference. And that was important so that after the human genome was complete, we could then study the effect of microbiomes on human health. And we needed a human reference to screen that stuff out. Well, everyone's probably familiar with the mice transplantation experiments where you can actually transplant obesity from one mice to its twin by moving the microbiome over. So it's very clear that this has a profound impact on human health, and we need to understand these genes. There are more microbial genes on your body than your own, but they're different genes. They don't have a lot of exon, intron splicing going on, so they arguably don't produce as many proteins, but there's still nevertheless more cells that are microbial in your body than human. So we want to pay attention to the microbiome. Now, fast forward to 2000 and, oh, I think I've missed a particular slide there, but if you fast forward to actually 2010 to look at cannabis genetics, I'm just going to step in here and see if I can resurrect that, you'll see something that doesn't look as if it is that modern. Let me see if surgery will prevail here. Well, it won't. So anyway, what we see today, if you saw the picture behind this, it was actually 2 people in a hotel room purifying DNA in Holland. Because in 2010, we didn't need a football field full of robotics. You could actually purify DNA from a species in a hotel room and get to its genome just by moving that DNA to a sequencer. Now, in the process of doing that, a few years later, we've been able to actually throw some of these robotics at the problem, and Ryan Lynch is going to be showing where these robotics actually lead to. I think I've got it going right there. Some of you saw the tour yesterday. So this thing is basically running around sequencing dozens of cannabis genomes to get us a better phylogenetic tree. And there's a good reason for this. But a lot of this effort is actually to drive and understand the genetics that drive cannabinoid expression and terpene expression, and perhaps a lot of other traits, perhaps flowering rates and oil and how quickly these plants might grow. We can perhaps tag those things to genetic variants and track them. And we're particularly excited about the changes that have happened since 2000, even the year 2000 when the human genome closed. The cannabis genome is not nearly in as good shape as the human genome. We have the human genome probably down to only 100 gaps right now, and the cannabis genome is far harder to sequence. It's probably the hardest genome we've ever sequenced. It's about 1 billion bases in size. It's 65% AT, which is a nightmare to sequence just from a chemistry standpoint. And on top of that, it differs by about 1 in 100 bases, whereas humans differ about 1 in 1,000. So this is the equivalent of having chimps being able to mate with humans and still have productive offspring. So this is a real challenge to sequence, and only recently have new sequencing tools emerged to help us on this. Now, one thing we did learn from the first time we sequenced this genome was that the THC synthase region of the genome was at 8x higher coverage than the rest of the genome. That stood out to us as perhaps being a gene replication event that was responsible for secondary metabolite expression, and maybe was also responsible for bringing us more THC synthase throughout the selection and prohibition. All right, but at the time, we actually couldn't properly sequence this because we only had 700 base pair reads and the gene was about 1,600 bases long, and we needed to know the perfect phase of this gene, which meant we needed to know which variants were resting on the maternal chromosome and which were on the paternal chromosome. There's 2 copies of every chromosome in this plant. And if you don't know that, and the gene happens to have lots of variation in it, it can be very difficult to call the amino acid sequence that's resulting from these genes. So we knew there was gene expansions. We didn't know what they coded for, and there was naturally a question, did these other genes code for these other cannabinoids? Because we really only see 2 genes in the genome that can code for cannabinoids, CBD synthase and THC synthase. Fast forward to today, and we now have these single molecule sequencers, which can luckily sequence 40,000 bases at a time. It's a real breakthrough. The challenge is they only have 85% accuracy. So to get the accuracy to a place where we want it to be, we can play some tricks with 40,000 base pair read lengths, as we can actually clone this DNA with some hairpins, and they call them smart bells, And this thing can then run around like a hamster on a wheel and sequence this thing around and around and around like a circle and give us a error-corrected sequence. This is what we call a circular consensus sequence. And it does this with these zero-mode waveguides. If those of you are familiar with microwaves, you may see a screen in front of that microwave. That is to basically make sure the radiation, which has a larger wavelength than the diameter of the whole of that screen, doesn't emanate through it. But what it does do is it leaves a little finite zeptoliter volume of a radiation of that wavelength at a pinpoint in the whole. And that pinpoint is so small that a single polymerase can fit there. And we can watch that single polymerase incorporate DNA signals in real time. Now, just due to the nature of loading these things, about a third of them only get loaded with a polymerase that can read, but nevertheless, there's millions of these holes. We can read basically the THC synthase sequence of dozens of cultivars at a go, and as long as we run it around 20 or so times, we get the accuracy up to a point where it is acceptable to understand the amino acid sequence of all of these things. So when we've done this on many drug-type cultivars, what we find is there's actually a lot of copies of THC synthase, and now we know the amino acid sequence of them, and we can determine which ones are active and inactive and which ones are still question marks. There are a lot of inactive copies that are getting replicated. These have frameshift mutations and they probably don't code for anything. There's a handful of other ones that have single amino acid changes that might have a different efficiency of folding the precursors, or they may actually have other side products that might result in these other cannabinoids folks have seen. But lo and behold, we still only see two major synthase-like compounds in some of their relatives, which we could argue maybe can account for about 10 of the cannabinoids and their derivatives. But we can't explain where all the other ones are coming from with just this alone, unless the side product thing is real. Now, the other literature that's out there are that microbiomes potentially transform these things by hydroxylating different parts of the side chain, and maybe even Dr. McKernan was recommending that they might hydroxylate some parts of the terpene precursors that would change the way that these things fold. Albeit most of these papers suggest it's at very, very low rates and yields that this occurs, and that they can't get enough of the material to do any type of clinical study with it. Nevertheless, this is very different than what we see in the terpene synthase pathway. The terpene synthase pathway in plants looks a lot like a plant or a vertebrate gene. It has lots of introns and exons, and we get all types of alternate splicing. And it's perhaps this alternate splicing that might, in fact, be governing the diversity that we see in terpene expression. So where did THC synthase come from? This is a very important question. There's a berberine bridge domain in this gene that we see in poppy, which folds opiates. And we also see it up and down the angiosperm line. Then there's some very weak homology to bacteria and fungi as well, and it kind of looks like a microbial gene. It has a start and a stop, and it's a single exon. Which is very interesting to us, considering some of the work that Dr. McKernan's published on quorum sensing with these compounds, and that they, in fact, might be antimicrobial. So these compounds look as if they are communication vehicles to the microbiome that might sit on the plant, and that could be very important to us when we're trying to understand this level of complexity of terpene expression that can occur with these multi-exon genes. I think we can understand a few genes could do this if they had all types of alternate splicing. It's harder for us to imagine 100 cannabinoids coming from just 2 genes with the diversity that's in there. But maybe that's what's in fact happening. So we need to know the microbiome then of these plants, and we set out to do is design a test that could be used in many of the states that are doing mold testing and bacteria testing. We're mandating mold and bacteria testing in almost every state that cannabis gets legalized in. However, the techniques that we're using to measure this stuff do not really identify what bugs they are. They just count colonies, and it's about a 100-year-old technology known as a petri dish. Now, culture is a very interesting thing. Getting things to culture has maybe only 70% or less of the microbes are culturable. Some argue it may be even lower than that. And on top of that, it's not very specific. Carbon sources don't give you the type of information that DNA does. So we designed a test that actually operates off of the ITS region of the genomes. This is the part of the genomes that puts you on the tree of life. So they're highly conserved regions in the ribosomal DNA operons that you can land primers on to specifically amplify one of these genera. And in doing so, there's information inside of here that if you were to sequence it, would give you high specificity in exactly what genre and species you're dealing with. So we designed PCR assays that target this region and give us quantitative information as to how much microbial content is present on the plant. And once we have that, if there's any questions of one that happens to be very high in signal, we can then go in and inquire what it is. And we do that by sequencing these PCR products, and it gives us a little microbiome idea of what's going on in these plants. So now we have a nice picture of all the bugs that might be on cannabis plants, And we can go through and itemize them and ask, okay, we see some aspergillus, we see some fusarium and some botrytis, but here's one Girl Scout cookie that has a tremendous diversity and a long tail of different microbes. And the other ones these ones are a little bit older samples, and they seem to be dominated by just a couple microbes. A couple of them stood out, however, because these ones we were not finding on anyone's radar in terms of being a problem in healthcare or on the plants. These are penicillium species, and they don't grow very well in traditional culture systems. They need different temperatures and longer time for these things to emerge. Lo and behold, when we look at these compounds, these microbes, and we try to grow them, we have a very difficult time growing them in just a couple days. These are 3M Petri films that didn't grow them, these are Biolumix cartridges that didn't grow them, and these are yeast and mold things from Simplate that didn't grow them. We can readily see the DNA. This naturally evokes questions whether the bugs are alive or dead, so we had to go and confirm that we could in fact grow these in the right conditions, and that if you ran the sequencing before and after growth, you could actually see a shift in how these things were growing over time. So they are live, we do know that, and they happen to produce these interesting compounds. Now, whenever you do ITA sequencing, it's standard to go back and confirm that with a secondary assay that it's really species specific and you've got the bug that you're thinking about. And so we targeted the paxilline gene that actually makes the paxilline and the citrinum gene that makes it. A whole genome sequence those genes after we amplify them and prove that they're in fact there. Now, what we don't know is how much they're expressing, but an important point is these all look very lipophilic and they will probably co-extract with CBD. They also happen to be, this one in particular is a nanomolar drug. So a small percentage contaminant of this can actually have a bigger impact than CBD alone because it's a potassium channel blocker, and it's been published to have contraindications with CBD in mice. We have another one here, citrinum. This one actually is known for a disease called rye grass staggers on farms. So some cattle can eat rye and have some of this penicillium on it, and they'll stagger around with some type of ataxia. This one is more of a nephrotoxic agent that's actually involved in a pathway that we know about from Roger Pertwee's lab and Duncan Ryan's paper suggesting CBD actually targets this region in the VDAC1 receptor in the mitochondria, and citrinum seems to be playing some role in there. We don't really know what role it is, though. So this set us on a mission with Digipath to try to see if we could detect whether the compounds are actually there. It's one thing to show the genes there. You've got to show that it's actually expressing the compound. This can be very difficult in the cannabis background. So we started with paxilline from Sigma and started spiking that into cannabis matrices to see if we can pick it up. And at first sight, what we can see is that we lose about 5 to 10-fold signal in trying to detect this if we use the Uhlig method that was described using an LC-MS-MS spectrum of this. When we spike this in, we lose about 5 to 10x signal, so we're still working on how to do this with the samples that we detected in a few slides prior, but we now have an assay, thanks to Sydney's group, that can detect this. We just have to make sure we have the sensitivity to pick up stuff that might be nanomolar in concern. So just to summarize some of this microbiome work, what we have here is we've seen that the culture-based systems are basically screening things on any number of colonies. Now, this is not a very accurate system, and we're quite concerned about this because we don't know what microbes are in fact there. As we mentioned before, 99% of the microbes in your body are commensal, symbiotic, and harmless, and we don't want to screen those out. We really need to get specific about the ones that are a problem and target the ones that are actually a human health risk and not just start screening out microbes that the plant might need. Our fear in screening out microbes the plant might need is very analogous to if you had to live on antibiotics. You might screen out microbiomes that you need to survive, and we may create an enormous genetic selection against certain genetics in the plant that can't make it through this very grotesque screen. So what we want to do is dial in and get much more specific with the DNA and hopefully fail less material and only point out the ones that have health risks. Now, what we've seen to date is we can already tell that most of the yeast and mold assays out there, when we see colonies on them, we grab those, we sequence them, and what we tend to find are bacteria that are native to humans and are not harmful. We see staph, we see Pseudomonas, we see acne, propion bacteria. Now, there are some Pseudomonas we need to worry about, aeruginosis, and we have seen that on occasion on some cannabis samples, but generally it's a ubiquitous form of Pseudomonas. It's not a concern. Likewise with the acne samples. So we need to be aware of this. The flip side of this coin is that a lot of bugs do not culture at all, and we've had samples that are totally loaded with mold that you can see with your own eyes, and they don't grow at all. DNA certainly picks these things right up. Okay, so this was published, and thanks to some people in the audience, John McPartland really deserves to be an author on this paper because he gave us such an incredibly constructive review, and I really like this mechanism of publication because of that, because the reviewers really actually do strengthen the paper. But this is a paper where you can review a lot of this work and see what we did to compare this to a variety of different culture systems. So I'm going to flip this over to human genetics, which is 5.5% of the human population are going to develop a genetic disease at some time in their life. And this is, it could go as high as 60% over time. And so we want to understand those genetics, because if we're going to be applying cannabinoids and perhaps some mitochondrial transformed cannabinoids onto humans, we want to understand the genetic diversity in the patients that this might apply to. So in doing this, we primarily have been focusing on mitochondrial dysfunction, and we did this for a reason. It does have some inroads into the ECS, and the reason this is a very productive place to start sequencing in the human genome is that most of healthcare is very skeuomorphic in that it is designed according to anatomy. We have cardiology, we have nephrology, we have neurology. These are all organ systems that we've built healthcare silos around. And when you're dealing with a dysfunction in the Krebs cycle, it is very agnostic to which organ it is in. It can bounce between any of these, and that creates an enormous expense in a healthcare system that likes to specialize based on organs. So patients that have this type of dysfunction can sometimes take 6 years to get a diagnosis and an enormous number of bills. And of course, this leads into seizures, because your brain is 2% of your body weight, but 20% of your energy consumption. So if you have a mitochondrial dysfunction, it actually can find its way into pain, into GI issues, into seizure disorders, and into ASD and developmental delay. So we've developed a lot of these panels to target these different regions. They're going to be spoken on a little bit later, but I think the really exciting work that we're seeing, and this is published by Bernard a few years ago, is that we are seeing a lot of CB1 localization to the mitochondria in neural cells. About 15% of the CB1 receptors sit there. So there's definitely connections, and there's something going on there. Now, why do we want to know this genetic information? We want to know this genetic information because we're finding a continually growing list of genetic contraindications or indications for particular genetic variants. Okay, so the sodium channel 1 mutation is probably the most well-known in this audience, because this is one that signifies Dravet syndrome. And although people in the past used to avoid sodium channel blockers, this might be a signature to actually move on or go directly to Epidiolex or CBD, because these patients are responding the best in those studies. We also have glucose transporter variants that are indicative of a 90% response rate with a ketogenic diet. So if you have that variant, you might as well go right to a diet change, and that may help with the patient. There's these polymerase gamma variants that you want to avoid with valproic acid. If you have this variant and you're given Depakote, you can destroy your liver, and you end up with a $500,000 liver transplant. All right, this is a very important thing to find out, and you want to know about it before you jump onto Depakote. There's other indications with GABA receptors and stereopental and calcium channels changing this, but I think the one that probably resonates the most, you may hear later, is that there's a vitamin B6 form of epilepsy, that if you have a mutation in this gene, you can treat the patient with vitamin B6, and you get a very good response rate. And this is a form of epilepsy that if you treated it with CBD, you might actually just mask the problem for 6 months and honeymoon out of it and realize you should have had B6 6 months ago. So that's one that's on our radar quite a bit. So how do we go about doing this? Some of you went on the tour yesterday. I won't spend much time on this. We basically have been sequencing panels and exomes, predominantly panels first in pediatrics, just due to the fact that when you sequence whole exomes, you can uncover a great deal of information about people that never consented to being sequenced. In other words, their relatives. When you find ApoE variants or BRCA variants in these pediatric patients, that implies some other family members may have some risk as well. And so ACMG prefers that you dial this into panels first and expand to exomes later. But the most important part of the technique that we're running up there, outside of the analysis that goes on, is in the laboratory. It's a very condensed laboratory, because every patient that we sequence, we hydroxymethylate their DNA in the active amplification, so that DNA is ephemeral and gets just self-destroyed in the process of sequencing. And that's needed so that we never contaminate someone else's DNA with another, and it makes the lab footprint half the size. This has been published in Nature Biotechnology. It has some implications for gene patents as well. As I mentioned earlier, this is the progression we tend to start with is maybe 500 to 1,000 genes that we'll sequence. We try to dial this into just the genes that are in the literature that we know are connected to the particular phenotype. And if we don't find anything there, there's a possibility of expanding to exomes. And the difference between panels and exomes is just the size of the box that you order these days. These things put out 250 gig in a shot, and these do about 10 gig in a shot. Now, inside this panel, there happens to be 39 different genes that we know are tied into the ECS. We have many of the sodium channels. We have the VDEC1 receptor in here. We can see CNR1, CNR2, FA, MAGL, DAGL, mTOR, and a lot of these other ones, particularly the P450s that are of importance. This is not all of them. We think the list is probably about 175 long, but we're not sequencing those other genes right now because it's tough to find any clinical correlation with a variant in those genes to an actual human patient. I think in time that will change. So when we do this, I want to touch just briefly, I think I have time, on the P450 system here. It is well known now that, that CBD is effectively like a grapefruit juice warning on a drug. Okay, so it is a potent inhibitor of CYP3A4 and 2C19, which means if you have drugs that have grapefruit juice warnings on them, grapefruit juice has bergamot in it, and that actually goes through CYP3A4 and inhibits it, CBD will slow down the metabolism of other drugs. Clobazam, or Onfi, is one that was seen in the Epidiolex trial, that this can actually escalate that in the blood. And this is a prodrug, so when you change the metabolism of a prodrug, you're slowing down the conversion of the drug into the active form, and it can lead to some complications. Same is true with carbamazepine. Both of these drugs, I would caution just to make sure that you have your HLA-1502 variant sequence, because they're both known to actually trigger Stevens-Johnson syndrome, which can have 30% mortality rate if you're not careful. Not very frequent in our population, very frequent in Asian and in the PAC-Rim populations. So P450s are important because to know this helps you understand how all of these drugs are going to bounce around with one another, and most patients with epilepsy are on 3 drugs, if not more. So what we do is we provide these tables of drug-drug interactions that if you are on CBD, what of the other drugs are going to go up and down in the blood according to the known literature, and of course which variants are involved in the metabolism of these, whether they're fast metabolizers or slow metabolizers, and this may help guide treatment optimization. Of course, we also sequence the entire gene, and that's important, not just nailing these SNPs, but when you sequence the entire gene, you might find frameshift mutations in that gene that are going to basically reverse if this is a fast metabolizer genotype. It's not a fast metabolizer if there's actually a frameshift next to these SNPs, and so that can be important information to capture with whole genome sequencing. And we do see that stuff on the panels. Okay, so I'm just going to wrap up with what you can do when you get to a pace of doing about 1,000 patients a month, like we're doing up in Woburn, is that serendipity will have it that you'll sequence some patients that are actually in current clinical trials. And so we tried to organize this a little bit better than serendipity, and we called up Oren Davinsky and we called up Eric Marsh and a few others and said, hey, can we sequence the patients in your study, hopefully to get to a common polymorphism that might predict response. There's been great success with Epidiolex out there, but there is a small subset of patients that get worse. And when they get worse in epilepsy, they can be intubated, you can end up in the ER, you can end up with status epilepticus. This can be very expensive, and so we want to see if we can dial that in. And what we have found to date is only 55 patients have been sequenced to date. And with those 55 patients, we're starting to see some signals, although I will caution you, and David Goldstein would chime in here saying anything to 10 to the minus fourth could be a synthetic association and you need more patients. But nevertheless, the SNPs that are coming out of this are implicating a handful of genes that may have a predictive value on the response to CBD. Now, what's interesting is the ones in yellow up here have some published links to anandamide, and that is actually enriched next to everything else that's in the panel. That the genes that are falling in as the top values here actually have published links. So that's very interesting, but we need to do more, and we just started on this doing more with MGH. We have an IRB study approved to sequence 55 more patients, and MGH, interestingly, at Elizabeth Thiel and David Friedman down at NYU, has been doing some work showing this N-desmethylclobazam escalation with CBD is real and traceable in the blood. We don't yet know if it's correlated with those genotypes, and that's what we're going to get at by sequencing these other 55 patients. There's also another 50 patients that we're bringing in from an IRB from University of Alabama, Birmingham. I believe some of them are here in the audience. Thank you for joining us. And we have an IRB that's also grant approved now from CHOP. They just received another grant to sequence 50 more patients. So in the end, we'll have another 155 patients. Hopefully that will be enough patients to decide whether this is real or synthetic, and we may have a marker that can help predict cannabinoid response and maybe teach us a little bit about what's going on with the esoteric nature of CBD. Now, to do this correctly, you need a team of genetic counselors, and there's something on the X chromosome about genetic counselors. I don't know what it is yet. That's going to require a bigger study. But without these people, we would never get any of this done. It's a tremendous team up there that has a real interest in personalized medicine. And there's a lot of external people as well that have been helping us on this, including internal people. I want to have some call-outs to Digipath and Cindy Orsner. She developed that LCM-SMS technique. Now, there's going to be some presentations a little bit later from Ryan Lynch talking about the work we're doing with the list exchange and genotyping a lot of these plants to get a better understanding of the drift in the plants. And of course, I want to call out Doug Smith, who's been doing a lot of the work on the GW study with Orrin, Eric Marsh, and Russell Sinedo. Peter Doherty with Roberto, who's been doing work on getting this to the market. Jessica, Yvonne, and Wendell and Lay, they're all here doing work on various aspects of exome sequencing and microbial genetics. Christina and Adrian. Adrian did a lot of work on CBD synthase and single molecule sequencing with us over the summer. Christina's been helping on exomes as well. Ben, John, and Vashist as well have all chipped in. And likewise, some of our friends at SC Labs have helped us getting very interesting genetics to sequence. So with that, I will leave it for questions.
For just a couple questions, then we'll break for coffee. But if we could take a couple questions for Kevin, if there are any, please go ahead. He will be available at the coffee break as well to answer any questions you might have. Any questions? Sir, over here.
That was a great presentation, Kevin.
Can you speak into the mic a little bit more?
I will speak into the microphone.
There you go.
There we go. Okay, so
Thank you, Michael.
So, with the interaction of the microbiome and its potential pathogenic nature in some cases, do you anticipate remediation or sculpting or remodeling of a microbiome for a particular cultivar?
So that's a very good question. It's early days, and we're hoping it may help in just perhaps biomonitoring in growth rooms and to understand if anyone has particular infections or blights, that this will be a helpful tool just to understand that domain. There is definitely literature on using bacillus to knock out botrytis, and bacillus is harmless, and that's interesting to us. I mean, I think it's an important point because when you look at food, if we take these tests and we go into the supermarket and start testing cheese, you get a lot of CFUs from cheese, but it's not harmful to you. You know, it's basically one big ball of microbes, and so I think we have to be rethinking the colony forming system because it's not giving us information with any specificity about what to do if you have high CFUs. But I think it's still early as to doing what you suggest. I'm hopeful we can figure that out.
Sure. Hi, Fred Vogelstein at Wired. What's it cost to have your genetic test?
So the genetic testing costs can vary depending on whether you're just doing the microbial sequencing up into whole exome sequencing, but I would say the gamut can spread from some tests that are $500 up to like $15,000 depending on how many genes you're sequencing. These are insurance reimbursed usually, and so with that there's some limits on how much can ever get back to the patient due to out-of-pocket expenses.
Thank you.
Okay, thank you, Kevin.